Osmolarity is the term that is used to describe the concentration of a solution. We express this in osmoles, which describes the amount of solute, such as NaCl, to solvent, such as water. The first diagram shows a solution that has a high concentration on the right side of the tube and a lower concentration on the left. When a semi-permeable membrane is placed between the two sides, the solvent will flow to the side with the higher concentration until equilibrium is reached. Here we will demonstrate a real-life example of this principle. In this video, the boys will represent water, or solvent, and the girls will represent the solute. Remember, solvent follows solute.
Let's get together, yeah, yeah, yeah. Why don't you and I combine? Let's get together, what do you say? We could have a swinging time.
Now that we understand osmolarity generally, let's discuss it with numbers. In this diagram showing NaCl as the solute and H2O as the solvent, the water flows to side B because that is the side with a higher osmolarity, or in other words, a greater concentration of NaCl. In this diagram, both sides have the same concentrations, but side B has a greater volume of solution. If we wanted to add an extra liter of water, how much of the liter would we need to add to each side in order to keep the osmolarities equal? This problem is worked out for you on the slide. We can figure this out by dividing the 15 liters on side A by the total 45 liters and multiply it by the 1 liter we will be adding. We do the same calculation using the 30 liters from side B. This calculation shows that we should add 1/3 of the liter to side A and 2/3 of the liter to side B in order to keep the osmolarities equal. If we add the whole liter of water to side A, 2/3 of the liter would pass through the membrane to side B anyway, so that equilibrium could be reached. An important note to make is that osmolarity is determined by the number of particles in a solution. Some substances dissociate in a solvent, which increases the number of particles. The examples here show that when glucose is placed in water, it does not dissociate, but when ionic NaCl is placed in water, it will dissociate, which increases the number of particles in a solution and therefore the osmolarity. To calculate the dissociation of different substances, we use what is called ionization constants. For example, when NaCl is placed in water, it dissociates, but not all the compounds dissociate completely. So for NaCl, the ionization constant is about 1.8, meaning that if 1 mole of NaCl were to be placed in 1 liter of solution, it would be about a 1.8 osmolar solution. An interesting fact is that the osmolarity of body fluids can actually be determined by measuring the depression of the freezing point. A 1 osmolar solution freezes at negative 1.86 degrees Celsius. Okay, let's bring out the scientist. In this lab experiment, the bag contains a high concentration solution and the beaker contains pure water. The bag is permeable to water, which means that the water begins to flow into the bag, and as it does, we see the volume inside the bag increase. So how does this apply to our body? Osmosis occurs all over our body, but it plays a particularly important role in the kidney. Every day our kidneys filter 180 liters of fluid, but 179 liters of that fluid are reabsorbed through osmosis, which means only 1 liter of fluid actually exits our body. Oh yeah, osmosis is important.